In this video, we will continue our discussion about this Hemica microfluidic device. So we have discussed in our previous videos how we can develop this Hemica microfluidic device. In this video, we will cover some of the results that are presented in the paper uh, that shows what are the applications on which what are the applications where one can use this microfluidic device. So this figure mainly discuss about the effect of the stiffness and the degree of confinement on the cell morphology and migration. The stiffness means that how stiff the gel is. The gel stiffness can be controlled by its composition. In this paper, they have discussed about PA, a gel that is made up of PA and water and one more compound, and the degree of confinement is defined by the channel width. The increase in channel width will lead to the 2D like confinement, and the decrease in channel width like around 4 to 5 micrometer will provide us the 3D like confinement. So let us start with this circularity measurement, or the morphology of the cells remains circular or it changes. So they have measured it at different channel width and also with different stiffness. So they have used three kind of stiffness for all the experiments mentioned in this figure, that is 8 kilopascal, 15 kilopascal and 21 kilopascal. And in different channel widths they have find out that the 8 kilopascal always has 8 kilopascal has the highest circularity in all the channel width that we can see here. So this means that cells maintains its morphology in the low stiffness, in the low uh, in the case of gels with lower stiffness. Thereafter, they also check the roundness. Apart from the circularity, they also check the roundness, which is the inverse of the aspect ratio. And there also they find out the similar results. That is, the roundness is more in the case of 8 kilopascal cells all across the different width that they have examined. And the other properties that they have measured about in terms of cell migration is the speed by which cell migrate, and they can see that it's a biphasic relation. That is, with increase in width, they see the increase in the speed, but at and it reaches a peak at around this 12 to 15 micrometer width of the microfluidic channel that we can see here. And however, the speed is highest for this at 15 kilopascal stiffness of the gel, and compared to the others gels that are studied, that is compared to the other stiffness, that is 8 kilopascal and 21 kilopascal. And after this 15 kilo, after it reaches its peak at 15 micrometer width, we can see the decrease in the speed. So this indicates that cell behaves at optimum speed around at 15 kilopascals. Uh, for in order to get the optimum migration speed or maximum migration speed, cell needs to have a tissue that has a stiffness of around 15 kilopascal and has a channel width of around or the width of between the tissues should be around this 12 to 14 micrometer. And then they check the solidity of the cells, and at this different channel width and also under different stiffness of the gels. Uh, they have find out that solidity of the cells at 15 kilopascals is reduced compared to the other gels, compared to the other gels of other stiffness, as it can be seen here, as it can be uh, evident from the results that stiffness at 15 kilopascal is reduced. So this indicates that uh, at 15 kilopascal, because of the high speed, there is a change in the shape or change in the morphology of the cells. And they check the persistence, at the different channel width, they can see that After this they can see that at high channel width, that is above 17 micrometer, they can see there is a decrease in the persistence or decrease in the persistence of the cells that are studied under gel that has a stiffness of 8 kilopascal, that we can see here. They are drastic reduce in the case of cell persistence. The persistence means that cell remains their own structure, or it is related to the solidity. And after that they check whether they check their channel guided or we can say guided migration through these jumps and this frequency of the cell at a channel center. So this guided migration usually occurs at high channel width. So they have examines the cell guidance at around 20 to 27 micrometer width, and also this 2D like structure is more than 22 micrometer width. And they can see that there is a large jumps that they have observed in the case of this low stiffness of the gels, that is around 8 kilopascal compared to the other case. And this is evident that cells do not follow this contact guidance path when there is a less stiff gels are there. And the jumps indicates that uh, how many times the cells pass their center line of the channel width. And this frequency of the cells that remains at a channel center is uh, also shows the same results that we have observed in the case of jumps, that there are around 0.3 uh, 30% frequency of the cells are 30% frequency of the cells at the channel center get disrupted compared to the other case that we can see here. And at a high stiffness of the cell we can see very less cells that get uh, dis disrupt from the channel center. So this indicates that with higher stiffness, the cells are most likely to follow a guided path compared to the low stiffness of the gel. So this results indicates the effectiveness of this Hemica, a microfluidic device to study the cells under different stiffness and confinement uh, condition to get the idea of cell morphology and migration within without using the in vivo animal models. So that's all for this video. If you need to understand more about this role of effect of stiffness and degree of confinement uh, and how to study this uh, parameters using Hemica microfluidic device, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.